والأربعاء برضه موجود. يوم الأربعاء، يوم الثلاث والأربعاء أنا موجود في الكلية إن شاء الله تعالى. فأي حدا بده يمر. بس يمكن بعد 9 بعد 10 إلا إذا حدا ما عنده إلا الساعة 8 باجي. اوكي. بس خلينا نحكي على الساعة 10 بكون موجود، وبعد 10 لو أنا مش موجود في المكتب بكون موجود في ممكن بالمختبر اللي هو الطابق الأول، حاطط خريطة على المختبر على الباب. questions by emails I don't prefer questions by emails. واللي بعت لي ايميلز هو لاحظ انه ايميلاتي او الريسبونسز تبعتي very short، يعني يا دوب yes no، نادر انه اكتب sentence. لكن as an exception يعني عشان انكم تقفوا عشان ما تضيعوا وقتكم تيجوا على الجامعه، اللي بده يبعث لي ايميل خلينا نحكي نعمل هيك سيشن ساعه، اللي بده يبعث لي ايميل I can answer خلال هاي الساعه. Okay, it's like office hours later. كأنكم جيتوا عندي على المكتب، مش عارف ايش هي احسن طريقه، يبعثوا لي ايميلات ولا مش عارف نعمل شات ولا whatever. انا عندي فيسبوك اكونت صحيح و I use it، بس الفيسبوك is just for friends and family. Actually حتى friends and family ما مش لايك لحدا. أنا بس بتابع اخبار على الفيسبوك اكثر من انه بحط صور ولايكات مش عارف. Okay, so فما بعرف يعني ايش نعمل شيء ما بعملش كونكت لحدا على الفيس. I'm not your friend by the way. يعني بدك تشبك معي على LinkedIn. I think a lot of you did in the past few months. LinkedIn is for professionals. Facebook for professionals. Okay? مش شيء personal تحط لي رحله رايحها وسوي وخش ونشر ومش عارف ايش. No, no, no. Just professionals. Connect على LinkedIn and connect with me. Okay, I'll be more than happy to do that. Facebook is really شيء very casual. فايش في طريقه هيك نعمل شيء كيف؟ زي وقت محاضره يعني في لا ايش بروجرام انه ممكن انه نعمل جروب شات او something. في طريقه؟ كيف؟ لا مكتب المبنى اساسا اللي فيه المشاركة، المكتب اللي فوق المشاركة أول شيء. أي خلاص ماشي، ابعتوا لي إيميلز. خلينا نحكي ساعة من خلينا نحكي من 2 ل 3 4 هيك بتكونوا مخلصين شوي بالبالغ، فبكون عندكم أسئلة. أوكي؟ طيب 2 ل 3 يوم الأربعاء ابعتوا لي إيميلز الأنسر إن شاء الله. Again, the way you study biochemistry, many students get fooled من خبرة بالbiochemistry. They think إنهم فاهمين، بس in reality ما بيكونوا فاهمين. It happened to me لما كنت أنا كمان طالب، دخلت على امتحان organic chemistry يعني فاكر إنه وطلعت النص. But it just happens to many of us. Try to study with someone. After you study by yourself, study with someone else and test yourselves. This is the best way. تعال عندي على المكتب يعني أهلا وسهلا تعالوا. I like groups. بحبش حدا هيك لحاله يجي. I like groups. لأنه بصير في discussion، بصير في حكي. ممكن أنت تسأل سؤال وممكن زميلك كمان يسأل سؤال. فهيك تكون المنفعة شاملة للكل، وأنا بستفيد منكم باي ذا واي، يعني الفيدباك اللي باخذه منكم بيساعدني. فمرات كثير يعني بخلص المحاضرة بروح على المكتب بعدل على السلايد على طول بناء على الفيدباك تبعهم. خلينا نحكي إذا اليوم الأربعاء 2 ل 3 إن شاء الله I'll respond to your emails بإذن الله. وstudy in groups. أي سؤال أي شيء بالنسبة للكالكوليشنز إن شاء الله راح تكونوا easy simple. ممكن يكون في two level calculations يعني بدك تحسب شغله وبعدين تحسب شغله ثانيه بس النمبرز are easy. بصير في تنسيق بينه وبين الدكتور ديانا على الاسئله بينه وعلى كله هذا فان شاء الله ما راح يكون في اي مشكله. اي شيء بحط لكم اياه على الاي ليرنينج كاناونسمنت تفضل.
دكتور ديانا حكت انه نجيب الحاسب.
سمعت اليوم الصبح ما حكت هيك ف I sent her a message عشان يصير في coordination كويس بينه وبينهم. Yes. So we will do that. تقلقش. يعني حتى لو هلا شوف لو في آلة حاسبة الكالكوليشن رح تكون صعبة. لو ما في كالكوليشن رح تكون سهلة. فأنا prefer إنه ما يكون في كالكوليشن. اوكي؟ نعم. بلاش ايش؟
يعني بقريت من 5
لوغاريتم اوف 5. طب شو رايكم تعملوه على الكالكوليتر لوغاريتم اوف 5؟ هلا قبل ما تفهم اول شيء لوغاريتم اوف 5 and you will remember it for the rest of your life. بس logarithm of 1 again is 0. Logarithm of 10 is 1. 1. Logarithm of 5. It's in between. 0.4 then. Okay? So you can easily estimate. حتى لو تطلع على numbers you immediately notice انه ايش الفاليو الصح. طيب ماشي. Yesterday we talked about isoenzymes. Yesterday we talked about isoenzymes. And we said isoenzymes are different enzymes, totally different enzymes. But one, they catalyze the same reaction. Name. They are produced from different genes. They have different catalytic activity. They have different modes of regulation. And they are distributed or expressed differently in tissues. And we talked about lactate dehydrogenase as an example. And we said that lactate dehydrogenase is a tetramer. It's made of 4 polypeptide chains. Each one of them, by the way, has its own active site. And it's made of 1 or 2 types of polypeptides. These are the H polypeptide and the M polypeptide. And you can have a combination. So you can have all H or all M, and you have everything in between. That is 3M, 1H, 2, 2, 1, 3. And we said that H stands for heart, M stands for muscle. So again, if you have the all H, this means that it's mainly present in heart versus the all M, it's mainly present in skeletal muscle. And we said that basically the all M lactate dehydrogenase likes to take pyruvate to lactate, so it metabolizes, so it does the anaerobic metabolism, whereas in heart, it does the opposite reaction, preferentially or preferably, meaning that it takes lactate to pyruvate. And the reason is heart does aerobic metabolism mainly, muscle does anaerobic metabolism mainly. So when heart converts pyruvate to lactate, it produces some ATP during this process, reaction. Lactate leaks out, it's transported out of muscle cells and blood, it goes to the heart. The heart uses lactate dehydrogenase to convert lactate back to pyruvate, so that pyruvate goes through the aerobic metabolic pathway. وحكينا عن regulation. Here's another example of isoenzymes. There are many examples by the way, but here's another example. We have 2 enzymes, hexokinase and glucokinase. Glucokinase is also known as hexokinase 4. Okay? Now, these two enzymes, and hexokinase is hexokinase 1, the one that we refer to. These two enzymes do the exact same reaction. They phosphorylate glucose to glucose 6-phosphate. That's what they do. Okay? Now, but there's a difference in tissue distribution. Glucokinase is mainly present in liver. And hexokinase is mainly present in red blood cells, as well as other tissues, but we'll focus on red blood cells. So now we have to think of the purpose of these enzymes in liver and in red blood cells. Why is it that liver cells have glucokinase and red blood cells have hexokinase? The purpose of liver is to provide glucose to other tissues. That's what it does. And if there is any excess glucose, it stores it in the form of glycogen. That's what it does. So it has a high level of social consolidation, solidarity, هيك تكاتف اجتماعي. إنه في إيثار. That is, if it has glucose, it checks on other organs. يا جماعة, does anyone want glucose? I have some. So it provides glucose to other tissues before it uses it. Now, other tissues like red blood cells really depend on metabolism of glucose for the production of energy, because that's the only source of energy. Especially that there is no mitochondria, or there are no mitochondria in red blood cells. So the only source of energy is that metabolism of glucose for red blood cells. Now, liver checks on other organs. If all are fine, then what it does is that it takes glucose, it phosphorylates it, and then it stores it in the form of glycogen. And it uses some for production of energy. So that's the purpose of these enzymes, or the purpose of glucose in general, in these two tissues. Now, remember something. When glucose is phosphorylated, it cannot be transported out of cells. ليش? Because there is a phosphate, and it's negatively charged. So there is no way of transporting glucose 6-phosphate out of cells. So glucose 6-phosphate gets trapped, imprisoned inside the cell. Okay? In order to transport glucose out, it has to be dephosphorylated. The phosphate group must be removed, and then it can be transported out. So for liver cells, what they do, they phosphorylate glucose only if there is excess glucose. It doesn't waste ATP just like that. It doesn't phosphorylate glucose and then it says, oh wait, I shouldn't have done that. جماعة الورق. I shouldn't have done that. I shouldn't have wasted ATP. Let's go back. Dephosphorylated. Remove the phosphate. No. Before it phosphorylates glucose, what it says is, well, let's see. Does anyone have enough glucose? Is there enough glucose in the body? If there is, then it phosphorylates glucose and it wastes or it uses ATP. So, if we look at the catalytic activity of these two enzymes, glucokinase and hexokinase, we would find out that they are different actually. This is how it looks like for hexokinase, really highly hyperbolic, just like myoglobin. And this here is for glucokinase. So they have different Km values. And the physiological fasting glucose level is this level right here. So notice that for when you have fasting, when you're like waking up early in the morning, and you have this amount of glucose in your body, what's really active is what? It's the hexokinase. It's not glucokinase. So in the liver, this enzyme is not active. So it doesn't really store glucose. It doesn't phosphorylate glucose. But red blood cells and muscles require hexokinase to be active so that they can produce energy. Okay? Is that clear? So if you eat lunch, breakfast, dinner, whatever, and the level of glucose is really high, now both of these enzymes are active, especially glucokinase, because now there is sufficient glucose in the body. Glucokinase can now be active. Glucose can be phosphorylated so that it can be stored. So now it makes sense. Okay, so the other thing is regulation. So not only that the catalytic activity is different, also regulation is different. So first, you see the km values for hexokinase, it's about 0.1 millimolar. It's very low, which means that the affinity is very high. And for glucokinase, it's 10 millimolar, so km is high, meaning that its affinity is low. And it makes sense. That is, only when the level of glucose is high that it can bind to glucokinase in the liver, can be phosphorylated, and it can be stored. But for hexokinase, even at low levels, still, this enzyme is active. The other thing is that for hexokinase, this enzyme gets inhibited by glucose 6-phosphate. The product itself inhibits hexokinase. Again, it makes sense. So red blood cells are not selfish. Yes, they need a lot of glucose 6-phosphate. They need hexokinase to be active all the time. But if they have enough glucose 6-phosphate, that is the product, they are not selfish. They keep some glucose for other cells, for other tissues. So they say, let's keep some for muscles and brain and all other tissues. Whereas for glucokinase, it doesn't get inhibited by glucose 6-phosphate, and it makes sense. Now, if there is a lot of glucose, it means that there's a lot of glucose 6-phosphate in liver cells. It doesn't go to waste. It gets stored immediately. Glucose 6-phosphate gets stored or gets converted to glycogen, or it gets added to glycogen. So it doesn't go to waste. So it should not be inhibited by the product, by the glucose 6-phosphate. Okay, can you see that? So there's logic behind there's logic behind having different isoenzymes catalyzing the same reaction in different tissues, having different regulations or regulatory mechanisms, and having different catalytic activities. Okay, so there is logic behind it. Any questions? Yes. No, it's not. No, it's not. Yes, no, it's not. Diabetes has to do with insensitivity of the insulin receptor. Well, type 2 diabetes. The receptor that senses how much glucose there is in the body becomes less sensitive. Okay, I know what it does. Information on the side. If there's glucose binds to the, if there's glucose, insulin level would be high. And the insulin receptor senses, the insulin binds to the receptor, sending a signal to the cell, there is enough glucose. So a transporter it gets on the membrane, allowing for glucose to get in the cell. Okay? So what happens here, for some reason, and no one knows why yet, the receptor becomes less sensitive. So insulin is there, glucose is high, insulin is there. It doesn't bind to the receptor, or it doesn't send a signal inside the cell, so the transporter doesn't get on the membrane, and glucose doesn't get into the cell. Okay? So it has nothing to do with phosphorylation of glucose. Okay? So this is the logic behind isoenzymes. Now, we can also regulate, enzyme activities. Now, that's another mechanism. And inhibitors are used to regulate enzyme activities inside the cell, as well as we can talk about synthetic inhibitors as well. Not only natural physiological inhibitors, but also synthetic inhibitors. Now, these inhibitors can be divided into two types. You can have reversible inhibitors, and you can have irreversible inhibitors. Reversible inhibitors are physiological. Well, they can be synthetic, but all physiological inhibitors are physiological. So what happens is that you have an inhibitor that binds to an enzyme, and it inhibits it. And it can be released. It's reversible. Now, irreversible inhibitors are mainly synthetic, pharmaceutical, drugs that you find in whatever. Now, these inhibitors, what they do, they bind to the enzyme, inhibiting the enzyme completely because they are irreversible. They bind and they do not get released. So, you have an irreversible inhibitor that binds to the enzyme, meaning that the number of active enzymes could be lower than normal, but not for reversible inhibitors. Now, so let's talk about these inhibitors and their mechanisms and what they do. Now, we have different types of inhibitors. Now we have what we call competitive inhibitors. This means that the drug or the inhibitor competes with the substrate in binding to the enzyme, in binding specifically to the active site. So there's competition between the two. If there is high level of inhibitor, it means that it wins the competition. It makes it hard for the substrate to bind to the active site. But let's say that we keep on increasing substrate concentration. There is still an inhibitor, but you increase the substrate concentration. Will you eventually get to Vmax? Yes, you should, because there's a lot of substrate. So basically what it looks like, so what you have here is that the enzyme can either bind to the substrate or it binds to the inhibitor. You can have a complex of enzyme inhibitor, or you can have a complex of enzyme substrate. Now, meaning that here we have an inhibitor that binds to an enzyme. This is the enzyme activity. Hyperbolic, Biochemistry. Now, if we add an inhibitor at a certain concentration, and we have increasing substrate concentrations right here. Now what happens is, eventually, you would get to Vmax. If you add more inhibitor, well, it makes it harder for the substrate to bind to the active site. So, if you add even more inhibitor, it makes it even harder for the substrate to bind to the active site. But if you keep on increasing the substrate concentration to really high levels, eventually, you should be able to get to Vmax, because now there is more substrate binding to the active site. So what is really affected is what? It's Km. It's the affinity of the substrate to the enzyme. So here you have Vmax, which is 100. Now half of Vmax, 50. So for the inhibitor without, for an enzyme without a competitive inhibitor, Km is right here. Well, what if you add an inhibitor at a certain concentration? Well, you get to the same half of Vmax. You go here, you go down, Km is getting higher. Well, go here with more inhibitor. Go there, Km is really higher as well. So the competitive inhibitor, Vmax is not affected, because eventually you will get to Vmax. But what's affected is Km, the affinity of substrate for an inhibitor. You would need more substrate to get to half of Vmax. Okay? So Km is affected. Now, we can draw it using the Michaelis-Menten plot, or we can do this as well. We can use the line-Weaver-Burke. Especially that the fact that with the Michaelis-Menten equation and be hard to calculate, as I said, Km. Like if we do the Michaelis-Menten equation, here's an enzyme without the inhibitor. Here, if we add the inhibitor, we draw the straight line. Now notice that this point right here is what? It represents Km. So both of these lines would cross each other at this point because Km is not different. But then eventually, what happens here is these are different points. What do these represent? Vmax. Now these are different now. This is much larger. Now remember something as well. These points are, these points here are, this point right here is Vmax, so Vmax is the same for a competitive inhibitor. But these 2 points represent Km. Remember, these 2 points are -1 over Km. Okay? So be careful if I give you a plot like this, and I give you these numbers, and I say, this point is 1, this point is, this point is -1, this point is -10. So do the calculations. Now Km would be higher if an inhibitor is added than if an inhibitor is not added. Okay? So you need to flip it and be careful with the minus mark. Now, there are other inhibitors that are non-competitive. These inhibitors do not bind to the active site. They bind at a place different than the active site. So there is no competition, and that's why we call them non-competitive. There is no competition between these inhibitors and substrates in binding to the active site. So the substrate can still bind to the active site. So immediately you would think, is Km affected? Km should not be affected, because the substrate can still bind to the active site. But the activity is lower. So Vmax is lower, because even though the substrate is bound, the enzyme cannot catalyze the reaction. So what happens here is that here you have enzyme plus inhibitor, and you add a substrate. Well, you can still have this complex right here, enzyme, inhibitor, substrate. So the substrate can still bind to the inhibitor, whether it is bound to the, whether it is free of inhibitor or if it is bound to the inhibitor, it can still bind to the enzyme. So Km affinity is not affected. Km is not affected. But Vmax is affected because there is binding, but there's no reaction happening. So here we have an enzyme without an inhibitor, hyperbolic again. Well, if you add a non-competitive inhibitor, Vmax is reduced. If you add more of this inhibitor, Vmax is reduced. More, Vmax is reduced. Further and further. But what about Km? It would still be the same. Here we have 50% of Vmax. Go down, that's Km. Well, here we have another Vmax right here. Okay. Go down, let's say go down to here, go down, same Km. You have more inhibitor, that's Vmax. Go here, half of Vmax is right here, go down, that's Km. It's the same value. So Km is not different. The affinity is still the same, but the Vmax is affected. Yes? لما احكي ايش؟ Yes. ايش بيكون سايت؟ regulatory site. Regulatory site. مش احنا حكينا انه enzymes have the active site and they can have regulatory sites away from the active site. Okay, so these regulators bind to that location, that spot, and they change the enzyme, the structure of the active site. Go back to when we talked about characteristics of active sites, and I said that active sites are small in structure or shape relative to the rest of the protein. And I said, well, what's important about the rest of the protein? They support the structure of the active site. So you can have a regulator and inhibitor binding at a place other than the active site. changing the structure of eventually the active site itself. So you can have the enzyme, the substrate bound to the enzyme, but there's no reaction happening. So you look at the Line-Weaver-Berg. What you have here is it's the same Km. They cross at the same point on the x-axis, but these two values representing Vmax are different. It's higher. It could be lower for the enzyme plus inhibitor than enzyme without an inhibitor. Remember that the y-axis is what? It's 1 over V. So if this value is 1, and this value is 10, and that's 1 over V, it means that now it is, it's different. It's 0.1 with the inhibitor, because you have to flip it. Okay? So this is the non-competitive inhibitor. Now, there's another class of inhibitors known as uncompetitive inhibitors, which is sort of like similar to non-competitive as a name, but the mechanism is different. So they're known as uncompetitive inhibitors. Now, what these do is that they bind to the enzyme only when these enzymes are bound to the substrate. So what they do, but there's no catalysis. There's no reaction happening. So if you have a substrate and you add these inhibitors to the enzyme, the substrate would really bind to the enzyme at high affinity, because you close your eyes, you open your eyes, and you have a substrate always bound to the enzyme, but there's no catalysis. So what does happen to Vmax? It's reduced because there's no enzyme reaction. Well, what happens to Km? It gets lower as well, meaning that the affinity becomes higher because you always have the substrate bound to the enzyme. So what you have here is without an inhibitor, it looks like this. With an inhibitor, it looks like that. So Vmax is lower than the enzyme without the inhibitor. How about Km? Km is higher. Affinity is higher. Okay, why? Because the inhibitor binds to the enzyme only when it's bound to the substrate. So Km is lower in this case. You look at the Line-Weaver-Berg, and it looks like this. Km becomes lower, but Vmax is Km becomes lower, yes, and Vmax becomes lower as well. Shit. Is that clear? Questions? So note the differences between these. So what you need to know here is the mechanism of action of these inhibitors, what they do, how they affect Vmax and Km, and you need to be able to interpret, to look at these plots, whether Michaelis-Menten plot or the Line-Weaver plot, and interpret what type of inhibitor we're talking about. Now the easy way is you memorize these. Now the hard way is to understand. Well, it's not hard, because you just need to understand. Questions? Okay, so let's go on with inhibitors. Now there are other types of inhibitors. Here we will focus on irreversible inhibitors. Inhibitors that are synthetic drugs. Now some of these inhibitors can be, these inhibitors in general, they bind tightly to active sites or to enzymes. They bind tightly to enzymes. They do so mainly covalently, and that's how they bind tightly. Now there are different mechanisms for these inhibitors. Some inhibitors are known as covalent inhibitors. Others are known as transition state analogs, meaning that they look like transition state molecules. And we have heavy metals. Now, remember the reversible inhibitors, what they do overall is that they decrease the concentration of active enzymes. So eventually what they do is that they inhibit Vmax overall. So there are different types of these inhibitors now. Let's talk about covalent inhibitors. Now, there are different types. We'll talk about some of these inhibitors, the common, famous inhibitors, and what they do exactly. Now, one of them is known as, is abbreviated as DFP, or diisopropyl fluorophosphate. This is the same as sarin gas, اللي هو غاز الأعصاب، الغاز اللي بيستخدموه بالحروب بسوريا، ومرة شفت التلفزيون ولا لا أي حدا عنده لما يكون في عندنا غاز أعصاب وبصوروا الناس، كيف بيكون شكلهم؟ Paralyzed and shaking. Exactly. Now, what happens with these inhibitors, with this DFP, is that they also use, some of these also they're used as insecticides. اللي بيستخدموه عشان رش المبيدات الشريرة للنباتات. Now, what they do is that they modify the active site of the enzyme acetylcholinesterase. That's an enzyme that exists in the brain, in the central nervous system. There is a neurotransmitter known as acetylcholine. This acetylcholine is important for the normal function of our brain. Now, but it should not exist in the vicinity, and it should not be present all the time around nerve cells, because you always have signal going in. It has to be inactivated. So acetylcholine is inactivated by an enzyme known as acetylcholinesterase. It degrades acetylcholine into choline and the acetyl group, and that's it. You don't have a signal anymore. This is the normal function of this enzyme. Now, what happens if we have these inhibitors added, if someone ingests or takes or inhales DFP, is that it goes into the active site of acetylcholinesterase and it modifies the serine in the active site. This means that this serine, and it's modified covalently. So it's modified covalently when it binds to the serine, and now serine in the active site cannot function anymore. So the enzyme is killed, it's gone, as if it's not there. So acetylcholine, the neurotransmitter, stays in tissue for a long time, causing the signal to be amplified. So that's what happens with these covalent inhibitors. Now, DFP can also inactivate other serine proteases, like chymotrypsin. But it's not really these inhibitors that cause the effects or symptoms of DFP.
هلا هو بهاي الحالة خلص الانزيم بيخرب ما في.
Yes.
طب ايش العلاج؟ ممكن يضرب كل الانزيمز تبع الديوفي صح؟ ولا لا؟
Yes, it will inhibit all serine dependent enzymes. في علاج؟ I don't think إنه في علاج. It's fatal. Another famous covalent inhibitor is aspirin, salicylic acid. What it does is that it activates, it modifies serine by adding an acetyl group. So this acetyl group is added to the active site, and the enzyme is inhibited. The enzyme we're talking about is the cyclooxygenases 1, 2, and 3. So this is the same mechanism, or the mechanism of action of aspirin. But other irreversible inhibitors can utilize another mechanism, which means suicide inhibitors. That is, they bind to the enzyme and they hardly get released from these enzymes. And some of them, actually, they look like substrates. Others look like transition state. Well, what happens is you have the substrate, or this drug, binds to the active site, and the enzyme starts the reaction. So what it does is that it takes a hydroxyl group from here to there, it takes an electron from there, down there, and then it takes another group and it says, okay, let's put it here, and it gets fooled. The drug does not have this extra group where you can have the transfer of a group or electron or whatever. So it gets stuck, meaning that the enzyme cannot go back and cannot go forward anymore. So meaning that the enzyme gets killed immediately. And that's why they call them suicide inhibitors. Now, examples of suicide inhibitors is a very famous drug known as methotrexate. It's famous because it's used widely in treating cancer. It's also used in reducing diseases related to inflammation. So like rheumatism, for example, joint pain. It's a chemotherapeutic agent. It has really severe side effects sometimes. It affects many cells. Now the way it functions is that it looks like a cofactor. An enzyme needs help by a cofactor. The enzyme we're talking about is known as dihydrofolate reductase. Dihydrofolate reductase uses this, the tetrahydrofolate, as a cofactor in the reaction. The reaction we're talking about is metabolism or synthesis of nucleotides. So during synthesis of nucleotides, tetrahydrofolate is needed to help the enzyme synthesize nucleotides. Now, methotrexate looks like this cofactor, tetrahydrofolate. Now what happens is that methotrexate binds to the active site of the enzyme, and the enzyme starts the reaction thinking that it is tetrahydrofolate, but then it stops. It cannot continue, cannot go on anymore. So eventually you have inhibition of synthesis of nucleotides, meaning that there is no DNA synthesis anymore. And DNA synthesis is needed for all proliferating cells. It's needed for all proliferating cells. And that's why methotrexate can affect all types of cells that need to be renewed, that need to be regenerated. Okay? Like, for example, hair cells, a layer of our gastrointestinal system. That's why cancer patients who take methotrexate, for example, they stay after taking methotrexate for like a few days, weeks, not able to eat anymore because there are no cells that are renewed anymore. Of course, the immune system is also affected as well because there's no renewal of immune cells. Another example of a transition state analog or substrate analog is the famous penicillin. Penicillin looks like the penicillin. What it does is that it inhibits synthesis of the cell wall in bacteria. Remember we talked about the cell wall in bacteria and we said how it's a peptidoglycan. So you have the repeated disaccharides, N-acetylglucosamine, and the N-acetyluramic acid. And we said that you have peptides associated with them linking these glycan sugars to each other, making the cell wall even stronger. Well, in order for cells to synthesize the cell wall, well, they need this diadenine peptide, this dipeptide right here. Now, penicillin looks like this dipeptide, and this adenine and alanine, this dipeptide is added to the cell wall. Now, penicillin looks like the substrate. So the reaction starts, but eventually what happens is that it looks different. There's a missing group, so the reaction stops, and that's what kills bacteria. They cannot synthesize cell wall anymore, and eventually our immune system can get rid of it. Okay, so this is penicillin. Now, we also have other irreversible inhibitors and heavy metals. So immediately when there's like a thermometer, mercury-based thermometer, and it gets broken, and you have mercury spilled on the floor, there's always an emergency taking place in that place. Back, mercury is on the floor. Okay, and they call security and whatever, etc., because it's very toxic. Lead is the same thing. الرصاص. It's toxic. لأنه what they do is that they replace the metals that bind to enzymes. Now there are metalloenzymes, enzymes that require metals to be active. So you can have certain again, you can have certain metals that bind to the active sites of enzymes, and these metals are important for the activity of these enzymes. You have mercury replacing these metals. So what they do is that they bind to sulfhydryl groups. Sulfhydryl groups, or the sulfhydryl group is present in which amino acid? Cysteine. Exactly. So all cysteine residues in active sites are modified. So the active site again cannot function anymore. Now lead, on the other hand, what it does is that it replaces the functional metal. So especially calcium. So enzymes that require calcium to exist in their active site, well, you have lead replacing calcium. And as a result, many of these enzymes become inactive, affecting many tissues, including the central nervous system. So this is how these heavy metals can function. Okay. So we have different inhibitors. Again, we have reversible inhibitors and we have irreversible inhibitors. Reversible inhibitors, we have what we call the competitive, the non-competitive, and the uncompetitive. And then the irreversible, mainly these are synthetic, like heavy metals, like the suicide inhibitors, or they look like substrates or substrate analogs or transition state analogs. And we have covalent inhibitors that modify the active site. Okay, but there's something different. Remember how we talked about immunoglobulins in different lectures? And we said that what immunoglobulins do is that they bind specifically to certain molecules. So their affinity is really high. Well, one scientist had an idea and he said, what if we make an inhibitor that looks like the active site? Sorry, what if we make an antibody that looks like the active site? So it binds to the transition state analog or transition state molecule. Okay? Again, so you have an immunoglobulin that is produced so that it looks like the active site, meaning that it binds to the substrate or to the transition state analog. What would happen in this case? What would happen in this case? It turns out that these enzymes can function like enzymes. They bind to the substrate and they can they can start a reaction. They can start transferring groups from one spot of a substrate to another. So they can really catalyze reactions. So this is something that is sort of like different than the inhibitors that we talked about before. Okay? There's another mechanism of regulation, which is regulation through conformational changes. That is, we change the conformation, we change the structure of the active site. Okay. حبيبي نام. طلع موبايلك، العب. أو اطلع. So we can regulate enzyme activity by changing its structure. And there are different mechanisms by which we can change the structure of enzymes. We have the concept of allosteric, and we talked about allosteric changes in the hemoglobin lecture. And we can change protein-protein interactions, we can modify enzymes covalently, or we can do proteolytic cleavage. So let's talk about allosteric regulation first. So allosteric, allo means different, okay, or other. Steri comes from the word structure, like stereoisomers, for example. Okay? So we're talking about different structures. And allosteric proteins are proteins that exist in different shapes, slightly different shapes. We're not talking huge difference in the shape of the protein. We're talking about small changes, like the 10 angstrom movement of the heme group in hemoglobin. Really small change, but eventually it changes the structure of the hemoglobin molecule by turning the peptides a little bit so that the electrostatic interactions are broken as a result of this movement. So this is allosteric change. Now, allosteric proteins in general, they are made of multiple subunits. So they are dimers, tetramers, trimers. They can be really large molecules as well. Really large, made of 60 subunits. Some of these subunits can contain the active site. Other subunits will contain the regulatory sites. So you have the catalytic subunits, and you have the regulatory subunits as well. Okay? Now, and what happens is that these regulatory subunits, they can bind to regulators, changing the structure of the whole protein a little bit, so that the active site, so that the catalytic subunit would have the shape of an active enzyme. So these are allosteric enzymes. Now, with these allosteric enzymes, they are regulated by different molecules. Now, they can be activated, or these molecules, modulators or modifiers, they can be inhibitors or they can be activators. So you can have small molecules that really activate the enzyme further, and you have molecules that can inhibit the enzymes. Now, these are known as negative modulators if we're talking about an inhibitor, or positive modulator or modifier if we're talking about an activator. And they bind to a place other than the active site. Okay? They bind at a place different than the active site, and it's known as the allosteric site. So what they do is that they bind to the site on a different subunit, and they can change the active type, the shape of the active site. Okay? Inhibiting the binding of the substrate to the active site, or they can change the active site, making it fit the substrate. Okay. So these are allosteric modifiers. Now, if the modifier is the same as the substrate, then we call this modifier a homotropic modifier. If the modifier is different, then we call it a heterotropic. So sometimes, again, you can have these modifiers as inhibitors, or they can be activators. They can be the same as the substrate, or they can be different than the substrate. So when we talked about hemoglobin, and I said that even though it is not an enzyme, but it's a nice example of allosteric proteins. And when I said that binding of one oxygen makes it easier for a second oxygen to bind, well, that's a homotropic effector or modifier. That is oxygen. And when we talk about allosteric enzymes or allosteric proteins as well, we're talking about changes in the shape of the structure, and these changes are gradual. So there is cooperativity, the concept of cooperativity. That is, you have binding of a molecule makes it easier for a substrate to bind. Binding of two molecules of these modifiers or activators can make it even easier for another substrate to bind to the other catalytic site and so on. So you have cooperativity. At the same time, you can have negative cooperativity, meaning that the binding of one inhibitor would make it harder for one substrate to bind. Binding of another inhibitor makes it even harder for the second substrate to bind, and so on. Okay? So there is cooperativity as well. Further. Non-competitive inhibitors work on enzymes that follow the Michaelis-Menten plot. أنا بنشوف البلوتس about allosteric enzymes. Well, in a way, it's the same concept that they bind at a different place, except the way the inhibitors function. They totally, non-competitive inhibitors totally inhibit the enzyme, whereas with these enzymes, their activity is reduced. Okay. you will see a few examples right here. The other thing is that we have, for these allostatic enzymes, you also have the T and the R structures, just like hemoglobin, the time or tau state and the relaxed state as well, just like hemoglobin. Now, what is the effect of these allostatic? What is the effect of these allostatic enzymes? What they do, well, there are two systems. The first important point is that notice that the shape of the plot is sigmoidal. It's not the mechanical, it's not hyperbolic. It is sigmoidal, just like hemoglobin, again, meaning that there is cooperativity. So whenever you see a sigmoidal plot, it means that there is cooperativity. Now, what happens with these inhibitors, they can follow one of two systems, known as the K system or the V system. With the K system, you have reduction in the Vmax, but there's no effect on K50 or K05, which is used as a description or reflection of affinity of a substrate to an enzyme, but it's different than Km. So Km, the Miccal is constant. This is reserved for non-allostatic enzymes. For allostatic enzymes, we use this term right here, K 0.5 or half or 50. So in the K system, the Vmax is affected, but not binding of the substrate to the K half. Or the V system here, what you have is Vmax is still the same, but the K50 is different. So you have two types of mechanisms by which these inhibitors can function. I'm gonna stop here and continue next week, inshallah.